Welcome back, friends. And we took an example as Escherichia coli to talk about the DNA replication in E. coli in more details. And we have a series of videos. In the first video, we talked about the initiation of DNA replication in E. coli. Now, this is the second video where we talk about the elongation of the DNA replication in E. coli. And it's not actually in E. coli but also in all other prokaryotic organisms. The elongation is kind of very similar, and also in eukaryotes the elongation is very similar with the eukaryotic and prokaryotic DNA replication. So what happens at the end of initiation phase in DNA replication in bacteria? You will see that DNA is melted, double strand DNA is melted by the application of DNA helicase enzymes. In case of E. coli, the name is DNAB, and then single stranded proteins are there, and one single stranded proteins are attached to the single strand of the DNA at the replication fork. Remember, replication bubble means when the DNA strands are separated, it forms a bubble like structure, where at the two end we have the replication fork, where the replication fork means the specific point of the DNA, specific region of the DNA, where you have a few stretch of double stranded DNA and then separated single strand of the DNA. That junction, that known as a fork because it looks like DNA fork. So here at this point, what we know that after the SSB is attached, then we recruit another protein called DNAG in case of E. coli. DNAG means it's the function of a DNA primase enzyme. Primase means it's nothing but a RNA polymerase enzyme which will add some ribonucleotide sequences at this start point, at the branch point of this fork. It will add some of them, and once it's adding the nucleotide sequences there, what it will do is that some of these SSB proteins will be separated. Okay, SSB proteins will be released from that place. Now the idea, let me draw a fresh model of this whole drawing to make you understand how this whole process will work. So the DNA, and we have a replication fork. We are not drawing the other part of it because the bubble means there will be another fork at this side. I am not drawing it. Now here, let's say here we have the primase enzyme attaching some nucleotide sequences. It produces a 3' hydroxyl at the end, and this sequence is a RNA sequence, remember, because it's added by RNA polymerase enzyme, and this sequence carries a 5', it's a 3'. So obviously on this complementary strand we have a 3', 5', so 3' here, 5' here. That is the complementary nature of the DNA. The strands are placed like that. Now this is the condition, okay, and in this condition the idea is this primer, the stretch of ribonucleotide sequences 5 to 10 or 15, this stretch of ribonucleotide sequences is known as primer, RNA primer here in this case. So this primer is added in both of those strands because DNA replication should be carried out using both the DNA strands simultaneously. So the primer is added in both of the strands. Here as well as the primer will be added someplace here. In this case the primer should be added somewhere in this direction. Okay. So the problem lies, there is a problem in this whole system. The problem is because the DNA polymerase enzyme will take care of the ultimate polymerization purpose in elongation, right? Elongation means simply nucleotide sequences will be added one after another and a stretch of nucleotide will be produced. Just like the old DNA strand, a stretch of new DNA strands will be prepared by stitching deoxyribonucleotide sequences one after another, right? And that's the job of DNA polymerase enzyme, and the enzyme we use here is DNA polymerase 3. Okay, so use DNA polymerase 3 So DNA polymerase 3 is recruited. But the problem, DNA polymerase 3 can only produce the DNA strand, it can only polymerase the DNA strand in only one direction, and that direction is 5' to 3'. There is the directionality of DNA polymerase 3 to action. The newly strand DNA will be produced from 5' to 3'. Okay. So if you consider this scenario, for this particular strand, we know that the DNA polymerase will easily elongate this strand in this direction from 5' to 3'. Without any issue, it can elongate the strand. Now on the other hand, on this other strand, we have a different, because of the complementarity of the DNA, we have different directionality, right? So to keep this replication going from 5' to 3', polymerase needs to elongate at this direction, right? So this bottom strand, polymerase should move in this direction, the top strand, polymerase should move in this direction. And we also know that DNA polymerase will produce both of the new strands almost simultaneously, working as a single unit, not actually single, two such units are joined together and they start elongating the sequence same time. So how could a DNA polymerase, single DNA polymerase, can load at this place and can go in two separate directions? It's not possible. One molecule, how could it go in two separate directions? It should go and move in a specific direction, in only one direction, to make this thing synthesize, right? To make new DNA strands synthesize. That is a major problem regarding the elongation of bacterial DNA replication as well as the DNA replication in eukaryotes. Both of them, DNA replication in prokaryotes and DNA replication in eukaryotes, struggle at this particular point. To solve this puzzle, they decide one of their strand to form a loop. Because if we form a loop, then we can turn this directionality, both of them at the same direction. How? Let's assume we maintain the directionality at this upper strand. So we keep this strand as it is, but we want to change the direction of the bottom strand towards this left hand side. So how would you do that? We simply make it a turn. Just make it a turn. So if you turn this, for example, let's say, let's do that like that. If you turn this by forming a loop, it's like a loop, right? If you're turning this, then what will happen? Let's say we form this loop and now we are continuing at this direction. So now we can go in this direction, right? Now it will be 3' at this side, 5' at that side. So this loop is very, very important. Loop is very, very important to form, because in this case, in this particular case, in this upper strand there is no issue. The DNA polymerase will be loaded, it can easily synthesize there. But in this case, it's not the case. We need to form a loop. But there is a problem forming this loop. You know what? Because this is the double stranded DNA. This region, the DNA is double stranded. So helicase is moving in this direction. So it's unwinding the DNA on this side. So as it is unwinding, the DNA polymerase is moving in this side. So the primase that we want to synthesize will be formed someplace here, right? Some in distant places, right? So as the primers are formed, because as it's moving in this side, try to remember this very, very carefully, this particular point. Let's assume the scenario now. While looping, this side and this side, try to remember, to make a loop, remember, to make a loop, the DNA has to be unwound several times compared with this strand, right? but that's not also possible, because as we are unwinding, we are getting same length of the strands in both of the cases. Okay, so what we are getting, and we, whatever part is open, we are priming at that point, right? It's not possible to prime here at first. Priming will start from this terminal ends. How and why? Because the loop is open at this site. DNA is closed. Let's say the DNA is completely closed. Now it's open, slightly open, for example. So when the primer will be done here, now let's say the loop is further open. So the primer will be found at the terminal, not at the closed region, right? So the primer will be formed at the terminal, just like in this case. In the other strand also, primer will be formed at this end. At this end. So the primer will form like that. Primer will form at this direction. So what will happen? A primer formed in this particular direction, primer formed in this direction. So that means this is 3 prime, this is 5 prime. So now how can you move in the same direction? Formation of loop. How the loop will be formed? You just take this and there will be a jump like that, loop like that. So simply imagine of this, take this strand and you cause a loop like this. When you take the strand and cause a loop like this, now this is the end point. Remember, this is the end point. And you take the strand and form a loop. This is the end point now. So the DNA is now in this hand. We have like this. And if this is the scenario, and this is the end point of the primer. So the primer started here like this. Just assume the situation. It forms this loop. So once the loop is formed, then what we have? We have both the primers in the same direction. Both the primers at the same direction. So now polymerase can load polymerase 3. Polymerase 3 can drag both of the strands at the same direction. Right? So as it is moving in the same direction, what will happen? As this strand, this other strand requires to be open quite a long, because we are starting it here, while in the other hand, to form this loop, because you cannot form a loop while the DNA is open in small amount. You need to have a long amount of DNA to form the loop. So while it start to form the loop, there is a lot of DNA synthesis already going on in this particular strand. This is why strand is known as the leading strand, because the DNA synthesis is very, very fast. It's going on continuously in this leading strand. So this is leading strand. And meanwhile, stretch of leading strand is synthesized, then this strand gets enough long DNA to form the loop. Okay? And then they will start adding nucleotide sequences. So meanwhile, at that time, what happens? Few stretch of nucleotide sequences, already new DNA synthesis is done in leading strand, while there is no DNA synthesis in the lagging strand at all at the very beginning. Right? So new strand is opening, this leading strand complementary strand is open, strand is synthesis. On the other strand, it's open and it's waiting to have a right length to form the loop. So during that time, what happens? In this lagging strand, they are adding multiple primers, because in each time single primer is not sufficient to drag it, because in this leading strand we don't require any other primer most of the time, because we can easily synthesize the sequence for a long stretch without any alteration, because they don't need to halt, they don't need to change, they don't need to form any loop. But on the other hand, the lagging strand, we need to form the loop, and meanwhile the time we need to form the loop, there is a time to put a primer at this end. So there will be a primer at this particular site. So it will be like this. This direction there will be a primer, and let's say this direction there will be a primer, and this direction there will be a primer. So the primers are continuously added here on this other end of the strand, because this is distant and the primer will be formed at close to this loop. close to this replication fork where it's open. So now what happens, in the leading strand the DNA synthesis occur continuously, while in the lagging strand now as it gets the 3' hydroxyl, it will start preparing the nucleotide sequences. But until where? Until it reaches the primer. Whenever it reaches the primer, the another primer, then at this point the DNA polymerase will leave, and then again it will capture from somewhere here. Okay, or capture for somewhere here in this direction. Actually, sorry, I draw it in a different way. In this direction it will be, in this direction. So what happens again, they will start adding nucleotide sequences in this direction, right? So every time it start adding nucleotide sequences, whenever it reaches the earlier primer, it will drop that part. Then again it will hold on to another primer and start synthesizing DNA. And again whenever it finds the DNA, a strand, it will drop it there. So the directionality here, the primer directionality will be this. So again it will start adding nucleotide sequence, drop. Again it will start adding nucleotide sequence, drop. So what happens in this case, in this strand, we have a primer and a nucleotide and a DNA sequence, primer sequence and a DNA sequence, primer sequence and a DNA sequence. So stretch of the sequence like this. While in this strand we have one primer sequence and a consecutive long DNA sequence, because there is no issue with this. But there is the issue because we need to form the loop, and the formation of loop creates the issue, creates the problem. That's why we need to deal with this whole situation there. Okay, so remember how the loop is formed and why we have fragmented DNA on this lagging strand. So you must write it, this is a lagging strand, and what happens in this lagging strand, I told you about a primer, then a nucleotide sequence, then a DNA sequence, like that. And on the other hand we have the parental DNA sequence. So this is the scenario, this is a primer and a DNA sequence. So once the primer and DNA sequence, between those two things there is a gap, not a gap actually, there is a nick. Okay, now this RNA should not be present in the DNA. While in this leading strand also we have a stretch of RNA sequence, in the lagging strand we have multiple stretches of RNA sequence. So those things should not be present there. So what we need to do at the end of elongation process, at the end of elongation, we need to cleave those RNA sequences out and fill the gap with the DNA sequence. Okay, so we cleave them out with RNase H. RNase H. We apply RNase H and it will take this RNA, degrade these RNA strands, so they form a gap, and that gap is filled with DNA sequence. Let's take another color, DNA sequence, but again they cannot join this sequence at the end. So there is a nick. The difference between a gap and a nick means, gap means there are no nucleotide sequences in the middle, that's the gap. Nick means all nucleotide sequences are there, but the backbone, the phosphodiester bond is absent between two nucleotides, that's the nick. So once this RNase H will degrade all the RNA content, then the DNA is added, the DNA synthesis is added using DNA polymerase 1. Usually polymerase 1 will synthesize the DNA, and then this nick is filled by the DNA ligase enzyme. So the nick will be filled by DNA ligase enzyme. Okay, so by this way it will be completion of the sequence. So parental DNA, new DNA. That's how the whole DNA sequences are originated. So that is the idea of elongation stage. Okay. Now one thing that is the biochemical thing about the polymerization process, I did not talk about it. So that thing is very easy actually compared with these complications. That's why I talk about this complication much. You know, those fragments that we find with RNA and the DNA stretch, we call them Okazaki fragments, right? And all these things, Okazaki fragment formation, why we require so much of difficulties, formation of loop and everything, all these things are required for only one problem, that is DNA polymerase cannot initiate replication from 5 prime to 3 prime. Cannot initiate replication from 3 prime to 5 prime. The directionality is 5 to 3 prime. It cannot go from 3 to 5 prime, and DNA is complementary. So if they want to synthesize both the strands at the same time, they need to deal with this situation. That is only if the cell thinks that they will only carry the DNA replication once at a time, then this problem may not exist. But cell definitely does not want that, because cell want to divide the DNA very fast, because the replication stages are hugely fast. They need to produce a copy of the DNA in very fast conditions, very less time, so that they can divide. You know, in E. coli, the time for E. coli cell division is 20 minutes. So in 20 minutes, all the protein production, the separation, the production of DNA content, replication of DNA content, separation of the DNA content to the daughter cells, and all these things are complete in the 20 minutes. So you know, it's very fast. So that's why they need to do it in simultaneous process, not only for replication, but they carry transcription and translation also simultaneously. So that the process can work very fast. Fasting is very much important priority in cell, because it's very important to always grow very fast, especially in bacteria. Now the process I told you about, the process regarding the biochemical process of polymerization. The idea is very simple, that we have a template DNA. This is known as template. Template DNA means the DNA which we use as a complementary strand to synthesize our new DNA strand, right? So in the template DNA, if we have A, for example, if we have A, so the new DNA or nucleotide that we will put will be T. If it's T, the new DNA we put will be A. If it's C, we'll put a G. This is the complementary nature, and that's how we should put. So how we keep on adding nucleotide sequences like that? The idea is, at the end of each nucleotide sequence, if you look at each nucleotide sequence, for example in this case a C and say G, whatever it is, in the 5 prime of it there is a phosphate, 3 phosphate, at the 3 prime there is a hydroxyl. The chemical nature of a nucleotide, at the 5 prime end 3 different phosphate groups, at the 3 prime end 1 hydroxyl group. Now the phosphate group that is present here, as you know, we can synthesize it from 5 prime towards 3 prime, right? So what we see here, in this G or guanine, we have a 3 prime hydroxyl left here. Okay, now let's say the upcoming nucleotide. What should be the upcoming nucleotide? It should be complementary to the G, so it should be C, cytosine. So take cytosine. If you look at cytosine, we have a 3 phosphate group at the 5 prime end, 1 hydroxyl at the 3 prime end. Now this 3 phosphate cytosine will come. Okay, so let me draw it. Cytosine with 3 phosphate groups will be there. So let's draw it little bit. Phosphate group, 3 phosphate groups are there, okay, and a hydroxyl group at the 3 prime end. So now what happens? This hydroxyl group that is present in the growing chain, it will attack the alpha phosphate. This is the alpha, beta, gamma, okay, that will attack the alpha phosphate. It's a nucleophilic attack, okay. So once it's attack the alpha phosphate, then this beta and gamma phosphates are released. We call them as a PPI or pyrophosphate, that is released, okay, and they will form a bond between this G and C. That bond is a single phosphodiester bond, okay, so we call it a phosphodiester linkage. So C will be placed properly. Right after that, the same way G will be placed, and so on. Nucleotide sequences will be placed one after another by this fashion. It will be a nucleophilic attack by the hydroxyl. Lone pair of electron of this oxygen will attack the alpha phosphate. As a result, beta gamma phosphate will be released as pyrophosphate, and this process will go on like that. Now as it is removing pyrophosphate or PPI, the two phosphate groups, this pyrophosphate is energy containing group. It contains energy. So if we break down this pyrophosphate, it will release some energy. Okay. So this breakdown of pyrophosphate will give us the energy to drive this reaction forward. That's how thermodynamically the DNA replication process is favored. Okay, that's how the whole process works. And not only that, but also DNA polymerase enzyme will be present there, will also facilitate this whole process to function properly, because DNA polymerase has a specific structure where three different domains. DNA polymerase has three different domains. The structure of DNA polymerase is looking like a hand. The polymerase 1, if I talk about it, like this right hand, if I close it like that, there is a thumb, palm and a finger, three different domains. This palm domain is the actual catalytic domain. Thumb domain is to position all those nucleotides properly. Finger domain is also to check the bonding pattern, this complementary nature. If they find complementary nature, then only they will put this nucleotide to the place. If not, they will not put it, because DNA polymers have to make sure that a proper nucleotide should be placed, right? So if there is a G, it should put a C. If there is A, it should put a T, right? That thing is maintained by this DNA polymerase enzyme. How DNA polymerase actually gets there, how DNA polymerase actually place the DNA, and they will check the new DNA with the old DNA, and how the new nucleotides are entered. I have separate videos on that. You can watch that video. But the idea of this series videos is to make you understand the actual process, not about the tiny details. We have separate video on those tiny details. You can watch them, right? I put the links in the description, so you can watch those videos to understand how exactly this DNA polymerase function. Okay, but for a whole, the idea is it has these three domains, and at the bottom of this palm there is a domain called exonuclease domain. Exonuclease domain means the domain which can proofread whether they add a proper right nucleotide or not. If they add a wrong nucleotide, then this DNA polymerase changes its conformation, so that, see, like from there to there, so they can place that wrong nucleotide containing DNA to this exonuclease domain, which can cleave some of the nucleotide sequences, and then again it reshifts back to the normal conformation to start adding new nucleotide sequences, right nucleotide sequences. So the DNA polymerase has a capability of proofreading activity on its own, and that increases the chance of that decreases the chance of mistake and increases the chance of better replication process. Okay, but still it ends up with mistakes, and that's why we have separate sets of DNA repair mechanisms to deal with those situations. Okay, so that in a sense is the DNA replication elongation in E. coli, and how in prokaryotes DNA replication elongation take place. Okay, in the last video we'll be talking about the termination of DNA replication in prokaryotes. That's also a little bit unique. Thank you. If you like this video, please hit this like button, hit the subscribe button here, and subscribe to my channel to get more videos like that. Thank you.